So yes, just to say I'm standing in for my supervisor, the newly knighted Munir Pir Mohammed, to give this talk on genotype guided warfarin. So can everyone hear me before I start? Brilliant, yes. So approximately 1% of the UK and the US population currently takes warfarin, and there's a 40-fold variability in the doses between patients. Warfarin is also one of the leading causes of all adverse drug reactions that lead to patients being hospitalized. Just to review the pharmacokinetics and dynamics of warfarin. So it's a racemic drug made of 2 enantiomers, and it's the S enantiomer which is about 3 to 5 times more potent than the R enantiomer. And the S enantiomer is metabolized by cytochrome P450, the 2C9 isoform within the liver. The target of warfarin's action is also in the liver, and it's this enzyme VCORC1, which stands for vitamin K epoxide reductase complex subunit 1. And this is involved in the vitamin K cycle. And when you inhibit that, you inhibit the production of reduced vitamin K, and therefore you inhibit the post-translational activating modifications of the clotting factors 2, 7, 9, and 10, and thus you inhibit the clotting cascade. So on your left, there's a histogram there which shows that when you look at the different doses of warfarin that patients take, it approximates to a normal distribution. Furthermore, when you then look at the incidence of major bleeding, as we've already heard, it correlates with INRs above the therapeutic range, so above an INR of 3. And as you go above 4.1, the incidence dramatically increases. The CT just shows a hematoma around a leg. So this is a recent review article, and we've already heard warfarin, as we can see with the major bleeding, has a narrow therapeutic index, but it also has this 40-fold variability in the doses that patients take, and this is because there are many factors that affect an individual patient's drug dose that they require. When you actually then look at the different factors which affect warfarin dose requirements, what's interesting is it's the genetic factors contribute the largest amounts of warfarin dose variability, and in particular it's common polymorphisms within the warfarin therapeutic target, the VCORC1, which contributes a quarter to the warfarin dose variability that we see in clinical practice. The second top factor are common inactivating polymorphisms in the cytochrome 2C9 enzyme, which is responsible for inactivating the S-warfarin potent enantiomer. This is just a Manhattan plot, which again illustrates the importance of these two genes and their variants. And what's interesting is when you look at the research, we can explain approximately 60% of warfarin dose variability, but that still leaves 40% unaccounted for, and recent research has showed that non-adherence perhaps contributes 8-9% to that. So there's still 30% or more that we do not know why that variability exists. So just to give a quick overview now of a recently published randomized controlled trial, which was asking the question, if genotype, so these genetic variants contribute a large amount to the warfarin dose variability that we see, perhaps if we take them into account, can we improve the time within the therapeutic range, and ultimately could we improve clinical outcomes of people on warfarin? And so this study was done by Professor Munir Pir Mohammed, and it was involved really in 2 main sites, UK and Sweden, with 454 patients. And they were randomly allotted into 2 different arms. Sorry about the science, but this schematic just demonstrates the standard clinical dosing arm, which is just as you would expect in the EU, where we use a loading dose for the majority of patients for days 1, 2, and 3, when these patients have just initiated their warfarin therapy because they've either been newly diagnosed with atrial fibrillation, or in this study they were eligible if they had a venous thromboembolism. And then after day 3, so day 4, 5 and onwards, the dose was modified according to local clinical practice. Compare that to the arm under investigation. if the clinician decided that patients required warfarin, then these patients received point of care testing. So a little blood sample was taken, it was run through a machine which would provide within 2 hours the patient's genotype for these warfarin pharmacogenetic genes, the VCOR-C1 and the CYP2C9. And then that genetic data was put into an algorithm which also took into account clinical factors which are established and known to affect warfarin dose variability. And that algorithm was used to derive a patient-specific loading dose regime for days 1, 2 and 3 when they initiate their warfarin. On day 4, the INR was checked just as routine, and then that INR was then plugged into a second algorithm, a dose revision algorithm, which modified the dose for days 4 and 5. And then after day 5, these patients in this arm just received standard clinical care for modifying their warfarin based on INR, just as in the control arm. The primary outcome measure was the time within therapeutic range within the duration of the study, which is 3 months. And compared to the control arm, the genotyped arm had a significantly higher time within the therapeutic range. Overall, the absolute adjusted difference was about 7%. You're still only looking at around 67% or 2/3 of the time these patients spend within the therapeutic range. When you break it down by month, you find out that it's in months 1 and 2 where the patients who are having the genetic algorithm compared to the control arm, they seem to benefit most in the first couple of months of dosing. And these are just a few secondary outcome measures. The one on your left demonstrates that with genotype-guided dosing, you reduce the incidence of INRs peaking over 4. And there's a couple more secondary outcome measures which demonstrate that if you did the genetic dosing, you could decrease the time it took to reach a therapeutic INR and also decrease the time it took to reach a stable dose. So overall, the genotyping decreased the incidence and time above an INR of 4, and it also reduced the number of dose adjustments required. That was published in the New England Journal of Medicine alongside this paper, which was also published in the same issue. And this was a very similar warfarin randomized controlled trial comparing genotyping to a control arm based in the US. And they found no difference at all between the genotype and the control arms. So the water is muddy. I
think they used a different algorithm.
Yeah, I'm just spot on. So. So this is another review which has just recently been written to try to help account for these differences in these two similar but large randomized controlled trials. Why did one show a benefit with genotyping and one didn't? And there's 4 main areas. The one was the algorithmic strategy used. I'm not going to go into the details on that. I'm happy to answer questions. The other was the ethnic heterogeneity, the control arms used, and the clinical relevance. So what was interesting is in the UPAC study, in the EU study, virtually all patients were Caucasian, but in COAG, they were far more heterogeneous. And fascinatingly, in COAG, black patients who received the genotyping actually did worse than they did in the control arm. And that really drums home the importance of looking at the ethnicity of your patient when considering genetics. And the reason for this is that the next slide is perhaps clearer, but the most common variants that were tested in these studies, so the STAR2 and the STAR3 variants, as well as the VCOR variants, they're much more prevalent in Europeans than African Americans. However, African Americans have many other variants which have been associated with their warfarin dose requirements, such as these STAR6, STAR8 and STAR11 haplotypes, and those weren't tested in these randomized trials. This is, again, it just illustrates the same thing, that when you look at warfarin variants and classify the patients who have maybe 1 or 2 as sensitive and patients who have 3 or 4 of these variants as highly sensitive, you see that African Americans have hardly any of these variants, whereas Europeans and particularly East Asians do. Then just quickly looking at the control arm, in the European study we just used standard clinical practice, whereas in COAG they used a clinical algorithm, which is certainly not standard practice. This is a algorithm, again, where you put in people's clinical data and it produces a recommended dose, but it didn't have any of the genetic factors in. Some have said that, well, in this case, then really we should be using a clinical algorithm because it showed no difference in coag compared to the genotyped arm. But then again, the clinical algorithm has never been tested in a randomized trial compared to standard practice. And actually, when you then look at the percentage of time within the therapeutic range in the control arms of the two studies, in UPACT, using standard practice, we've achieved 60%, whereas in COAG, using this clinical algorithm, it was only 51%. So at this point in time, I wouldn't recommend using a clinical algorithm. So who actually benefits from genotyping? If you don't have any variants in these VCORC1 or these CYP2C9 genes, then actually there is no benefit over normal clinical practice for dosing. Really, it's the people who have two or more of these variants who's got a significant benefit in terms of their time within the therapeutic range. And that reached an adjusted difference of over 10%. It was 11.05. And 10% is a clinically relevant improvement. In the relay data, which was a trial of warfarin and dabigatran, 10% improvement gave to a 20% improvement in the composite clinical outcome of stroke, embolism and bleeding. And in an earlier study from the UK, again, there are clinical sequelae each time you have a 10% increase in time outside the INR range. This is a recent publication from the Lancet, which again adds further evidence to the idea that it is these people who have multiple variants in these warfarin pharmacogenetic genes which really stand to benefit most from not just using warfarin standard dosing practices, but using some other method of management. And they did two things. They took, first of all, 4,000 patients within this large randomized controlled trial of warfarin compared to edoxaban. And in those 4,000 warfarin patients, they showed that, again, it's those patients with multiple variants compared to the warfarin patients who had no genetic variants that were at a significantly increased risk of bleeding. And that bleeding risk, due to the genetic variants, added information beyond the HAS-BLED clinical score. Secondly, what they then did was they then compared the edoxaban to the warfarin patients, and they found that although overall the edoxaban patients, I think, overall had a slightly decreased risk of bleeding, the majority of that benefit was in patients who had multiple warfarin pharmacogenetic variants. So that leads to the idea, perhaps if we genotyped patients and they have no variants, they're safe on warfarin, but perhaps if they have 2, 3, 4 variants, we should be either using a clinical algorithm or just giving them a NOAC in the first place. This is a cost-effectiveness slide. It shows a cost-effectiveness acceptability curve, but the take-home measure is the point estimate, or the ICER, which is your incremental cost-effectiveness ratio. And NICE, as we all know, generally uses a threshold of £20,000 per QALY, and it appears that in the UPAC study, the genotyping was below this threshold and therefore could be regarded as cost-effective. So very quickly, just looking at the NOACs, there's potentially 4 NOACs, and this is excellent. It gives both prescribers and patients a greater diversity of choice for their anticoagulation choices. We've already heard about the advantages and disadvantages, and as we've mentioned, although there's no antidote, there are actually antidotes now available, or rather in the late stages of development for dabigatran, I believe apixaban as well, I think. But I think there's probably there's still difficulties trying to work out which oral anticoagulant we should give to one patient over the other. And for example, when you look at dabigatran, the fact that it has been contraindicated in mechanical heart valves, and the fact that bleeding risk has been associated with dabigatran plasma concentrations, that sort of suggests that perhaps, at least with that drug, there could be a role for monitoring akin to the INR with warfarin. So it's still not completely clear how we should go forward. This just illustrates that, as expected, NOAC prescribing is increasing within the UK. So to summarise finally, we've seen that warfarin has a narrow therapeutic range. There are multiple factors which affect its dose, but the single factors which affect the dose variability, the most of those genetic factors, and therefore we need to have strategies beyond standard clinical practice for tailoring warfarin dose to patients. The genotyping in UPAT was cost effective, and actually my prof is doing a implementation study with the North West Coast Academic Health and Science Network to put one of these point of care machines either into the Royal or the Countess of Chester, which can give us the patient's genetic data within 2 hours. And in terms of stratification, well, it might well be that patients who have these warfarin pharmacogenetic variants, they should perhaps be prioritised for a NOAC. Thanks very much.